Hi and welcome to IBspectrum. Today we will be looking at DP Biology Unit 1. In this video, we will cover cell theory, unicellular and multicellular organisms, stem cells and their applications. So let's get into it. Cells form the most basic functional unit of life. The cell theory has three primary postulates. All living organisms are composed of one or more cells. A cell is the smallest functional unit of life, and new cells are formed from pre-existing cells. This forms the foundation of several biological processes that are integral for survival. As with most theories, the cell theory also has some exceptions. The first being striated muscle fibers. These are muscle fibers that are fused together. They are very long and can go up to 300 plus millimeters. Additionally, since these cells have been fused together, they are multinucleated, which means that they have over hundreds of nuclei. That is specific to this case. Acetobularia, also known as giant algae. This is a unicellular organism, hence contains only one nucleus. It is, however, extremely long, about 100 millimeters. The cell theory states that cells are the smallest unit of life. However, that is being challenged here. Aseptate fungi hyphae. Breaking down the words, we can get a clearer picture as to why this organism challenges cell theory. Septa is the internal wall, which ideally compartmentalizes the organism, allowing for the hyphae, which are thread-like structures in the fungi, to be discrete. Aseptate suggests that there is no internal wall. Hence, this organism does not have separation of the hyphae and forms a continuous cytoplasm, which is multinucleated. This challenges the idea that living organisms are formed from distinct cells. However, in this case, due to the lack of septa, the organism appears to be one cell. Let's look at a past paper question. Which living structure is an exception to the cell theory? The answer is A. We determined that the answer is A since we just discussed it. However, it is also useful to understand why the other options are not viable. B does not specify giant algae hence we rule that out since the other types of algae that are not giant do not challenge cell theory. C is ruled out as the synthesis of organic compounds to form a molecule is not relevant to the question being asked with regard to cell theory. In fact, this is a concept that will be covered in Unit 2, so be on the lookout for that. D is ruled out as it describes differentiation, which is a normal cell process with the exception of stem cells, of course. They are undifferentiated and can undertake a variety of roles, and we'll be getting to that soon. Let's look at using Mr. H. GREN in Paramecium and Chlamydomonas, which are unicellular organisms. So you can recall the function of life as Mr. H. GREN, and then we can compare the two. So metabolism. As for Paramecium, enzymes catalyze chemical reactions with the cytoplasm, and this is true for both. Response. Paramecium has a response when subjected to stimulus, as does Chlamydomonas. However, as for Paramecium, upon touching a solid object, it reverses direction. As for Chlamydomonas, it uses its eye spot to determine where the light intensity is greatest, or the brightest light source. With regard to homeostasis, Paramecium and Chlamydomonas both have their internal conditions maintained. An example for Paramecium would be expulsion of water through contractile vacuoles. As of growth, Paramecium and Chlamydomonas both experience growth in size. Size increases in Paramecium as matter and minerals from food are obtained, while for Chlamydomonas, photosynthesis and mineral absorption result in increase in size and dry mass. For reproduction, Paramecium follows asexual reproduction through mitosis, as do Chlamydomonas. Excretion. Waste products of metabolism are removed in both cases. For Paramecium, CO2 from respiration is an example, and for Chlamydomonas, O2 from photosynthesis. For nutrition, Paramecium uses endocytosis, which means it ingests smaller organisms in vesicles, and Chlamydomonas are autotrophs, which means they produce their own food by photosynthesis. Multicellular organisms. It is important to understand the concept of SA to volume ratio, which is surface area to volume ratio, with regard to multicellular organisms. The surface area of the cell determines the rate at which materials enter the cell and leave it. The volume of the cell is essential for understanding the rate at which materials within the cell are used up or produced. However, both of these do not increase at the same pace. Volume increases faster than surface area. Hence, materials start accumulating and are unable to exit, because production is happening quicker than they are able to exit. Hence, new materials are also unable to enter the cell. This is why large organisms often benefit from being multicellular, meaning they have many cells. This also allows for cells to become differentiated. That is, they can be assigned certain roles to fulfill in the body. Differentiated cells are those that have been specialized to perform a specific function. For example, when production of red blood cells is occurring, the genes that are needed for making hemoglobin are the only ones expressed, despite the cell having the entire genome. Okay, let's look at stem cells. So, stem cells are undifferentiated cells. Hence, these cells have not been specialized, meaning they're free to perform a variety of functions since they're not committed to a certain pathway. The ability of stem cells to differentiate can be described as follows. Totipotent. They can differentiate into any cell type present in embryo and placenta, for example, the zygote. Pluripotent, which means that they can differentiate into any cell type present in embryo but not placenta. Multipotent, which means adult stem cells with the ability to differentiate into related types of cells. For example, bone marrow cells have the ability to differentiate into blood cells. Unipotent, which means they can only differentiate into the same type of cell. For example, heart muscle cells, also known as cardiomyocytes, they can only produce new heart muscle cells. Different sources of stem cells. So, stem cells appear in different parts of the body, for example, the organs in the tissue. This includes the brain, blood marrow, bone marrow, muscle, skin, heart, and liver tissues. Although it seems like stem cells can be easily harvested, the age and volume of stem cells plays a vital role in their ability to reproduce and function. So we look at some places where stem cells are found. The embryonic stem cells are the ones that are found in large numbers during the gestation period. They're the smallest stem cells discovered so far, along with the ability to become any type of cell in the body. Cord blood, cord tissue, they have a quality of being the purest and youngest stem cells you can collect. They are known to function more quickly and effectively than adult stem cells from other sources. Placental tissue, acquired from umbilical blood, these are stem cells that are obtained from these sources, which are known as umbilical stem cells. They are known as a rich source of the same type of stem cells found in the cord, blood, and cord tissue. Bone marrow. Stem cells harvested from bone marrow are rich in blood forming stem cells, which means they can form all types of blood cells. So these were the important ones. So now we can look at the ethics in stem cell research. So the majority of scientists within the field of stem cell research believe that pluripotent stem cells are essential to gain basic knowledge about how stem cells grow and develop so that they are the building blocks of knowledge that we hold right now. And it's a base of knowledge that we are going to acquire in future years. However, there are a few particular uses of stem cells that are extremely unethical, such as the human embryonic stem cells, since it requires the destruction of human life in the process of its research. HESC is also highly unethical because it has a very high rejection rate, making it not only a consciously wrong method of stem cell extraction, but also unreliable and risky. Other important ethical issues related to informed consent of both donors of gametes and embryos, as well as recipients of stem cells and cell products. However, the biggest advantage of human embryonic stem cell research is that it can differentiate into cells of all three germ layers, which is very important for further research in many other fields. So the question lies, is it really worth it? Okay, so we're going to look at the therapeutic uses of stem cells. An example here is leukemia. So leukemia is a form of cancer and it develops as a result of mutations. Leukemia involves the production of excessive white blood cells, and this type of cancer does not cause generation of tumors. The physiology of the disease is such that when WBCs are produced in the bone marrow, they are released into the bloodstream regardless of whether the person has leukemia or not. These excess WBCs accumulate in the bone marrow and the bloodstream, such that they outnumber healthy blood cells, therefore obstructing the functions of the cells. So, in order to treat leukemia, the excess WBCs must be killed, and this is done using chemotherapy or radiation. However, this also diminishes the bone marrow's ability to produce cells, since cells cannot divide once they've been killed. Hence, stem cells are introduced in the bone marrow to ensure long-term well-being of the patient. Therefore, before chemotherapy is done, the stem cells in the fluid of the bone marrow are extracted and stored. So once all the cancer cells are killed, the stem cells are replaced in the body to continue normal function. Stargardt's disease. So Stargardt's disease is also known as Stargardt's macular dystrophy, which is a genetic condition which most likely occurs due to the mutation of ABCA4 gene, which is responsible for causing a protein which is vital for active transport in the retina to become defective. This ultimately results in vision loss. Embryonic stem cells have been used as a potential treatment option for patients of Stargardt's disease. Essentially, the embryonic cells are converted to retina cells And this method has been tested on mice with a condition similar to that of Stargardt's. And the cells that were injected were accepted and were not found to be carcinogenic. Another condition where stem cells are used is tissue regeneration, which is probably the most important use of stem cells. So, doctors use stem cells from beneath the skin surface to make new skin tissues. And this is most commonly used to treat severe burns, where tissues that have been generated are grafted onto the damaged skin, and this allows new skin to grow back.